chapter 5, and we're going to begin with talking about genomic imprinting. And this concept really is isolated to autosomal genes. So, both, just keep in mind that when it comes to autosomes, we're talking about non sex chromosomes. And through the process of meiosis, the genetic material coming from the male and the genetic material coming from the female are going to make an equal contribution to that new generation. So it's theorized that um, the offspring will actually have equal effects of maternal and paternal DNA upon the phenotype. So, however, that is completely theory. It's not true for every situation, and we now know this because we've been able to track some genes that will only be expressed if they're transmitted by the male parent or the female parent. So, one of the classic genomic imprinting conditions is Prader-Willi syndrome, and these individuals will have very small hands and feet, sorry, not feet, and they'll tend to be pretty short in stature, have a variation of mental retardation, and be very obese. And the big reason for these phenotypes is because these individuals have a small piece of chromosome number 5 that is missing. So a deletion has happened. So what makes this interesting, in that it's not just a chromosomal thing like we talked about in chapter 8, but it's actually really dictated by the father. So uh, chromosome 5, if you get the version that is inherited from the father, then chances are you're going to have an individual with this syndrome. A second syndrome is Angelman syndrome, and these individuals have a different phenotype. They have uncontrolled laughter and muscle movement, tend to have a very large mouth, and can actually go through various severity seizures. They also have a small piece of chromosome 5 missing. It's a different area than Prader-Willi syndrome, and this condition is actually inherited from the mother. Genomic imprinting symptoms really depend on which parent provided the deletion. So with Prader-Willi and Angelman syndrome, there's lots of variation in those phenotypes of the offspring. So some of the other genomic imprinting conditions that we see a lot of today are bipolar disease, as well as carcinogenesis, and a lot of others. There's actually been 25 different conditions that have been identified as being genomic imprinting. So fetal alcohol syndrome is a big one. That can actually alter the gene expression in an offspring if the mother consumes alcohol during pregnancy. So here's a good table in chapter number 5, and it really lists out and gives you guys a good list of the different phenomenon that we've talked about. So whether you get a trait that's sex-linked or sex-influenced or sex-limited, and then the genetic maternal, cytoplasmic, and genomic imprinting conditions as well. So spend some time on this table before you study for the final, trying to get a good handle on which each one of these are. And then note an example of each individual phenomenon as well. Anticipation is one of the two last concepts that we're really going to talk about, and this is where in conditions with myotonic dystrophy, we really see anticipation take place. So this is an autosomal dominant muscle disorder. And anticipation, we've talked about it a little bit in previous chapters, but this is basically where the symptoms are going to get stronger from one generation to the next. And the reason that we see this type of anticipation happen is because a region is going to be really unstable in the DNA, and that can actually increase or decrease the size as the gene is passed from generation to generation. So what may be 5 kilobases in the father may end up being 15 kilobases in the next generation. So, with myotonic dystrophy, the age of onset is a big issue here. So, it's highly variable, as well as the severity of the disease. And those two different traits are going to be correlated with the size of the unstable region. So, if you have a bigger region, you're going to have an earlier onset. If you have a smaller region, you have a later onset. Here's just a few other diseases that have anticipation like qualities. One of the big ones that's becoming more um, diagnosed today is actually Crohn's disease. All right, so let's talk about gene interaction because genes don't necessarily solely act based on their genotype. The environment can have some sort of influence on them as well. So we actually see this example, and this is not the first time you guys have seen this, to where a genotype can have several phenotypes depending on the environment. So a classic example is how warm a plant will grow at. So if you have an environment that is at 20 degrees, then you tend to get a really small plant. If you get an environment that's at 50 degrees, you get a medium plant. However, if you have warmer environments, you tend to get bigger plants. So the environment really has the ability to influence the growth rate, even though they all have the same genotype. Another example that we've talked about as well is the Himalayan rabbits. So if the rabbits are reared at less than 20 degrees Celsius, then they're going to have black points, but if it's above that, they're going to have solid white phenotypes. And Drosophila wings are also an example of environment as well. So depending on what environment they're raised in determines how big their wings are going to get. So the colder the environment, the smaller the wings. The larger the environment, or the hotter the environment, the larger the wings. All right, so some of the continuous characteristics that we've been talking about in terms of traits are going to be things that we can measure. So this really gets back to the statistical aspect of these different traits and being able to assign a trait type to it. So there's really continuous characteristics and discontinuous characteristics. A discontinuous characteristic is a trait that just has a few very easily distinguishable phenotypes. So this would be something like carcass quality grade. So to where you had just a few possibilities. You have a prime ribeye, a choice ribeye, a select ribeye, and a standard ribeye. Just a few different categories that each carcass is going to be categorized into. However, a continuous characteristic is going to be displayed in continuum. So this is, you can think about this in terms of height, the growth rates, weight, etc. So somebody can be 5 foot 5.2 inches tall. A calf at weaning can weigh 517 pounds. So they're continuous traits that can be measured in a continuous fashion. So some of these continuous traits we also term quantitative traits, and these traits typically are going to be determined by a number of loci. So this means that really there's a lot of genes that are playing into the phenotype that we actually see. Weaning weight is a really good example because there's thousands of different genes that go into the trait of weaning weight. It's not just environment. It's not just one gene coming from the sire. It's not just one gene coming from the dam. So an easy way to determine the number of possible genotypes is to identify or to use the 3 to the n power equation. So if you have eight different genotype answers or genotype possibilities, 3 to the 8th is 6,561 different genotypes. So these continuous traits may also have differences among the phenotypes that will be very, very slight. So that's where we get into on carcass traits like a choice minus choice or choice plus. So they appear continuous, but they really kind of run together as well. So these polygenic characteristics are also termed continuous traits as well. Poly means many, so polygenic is where many genes play a role in determining a trait's phenotype. Polygenic characteristics lead into a couple of different genetic relationships. One is polygeny, and this is where many genes play a role in determining the trait's phenotype, so that would be like weaning weight. And then the other side of that coin is a pleiotropic characteristic, and pleiotropy is where one gene controls many traits. So coat color, in red Angus and black Angus cattle is controlled by one gene. The horn trait in cattle is controlled by one gene. Pleiotropy, a classic example of this in human medicine is the PKU gene. So we talked about this in chapter 1 a little bit, this phenotype. And this is where an individual will have a homozygous recessive genotype of little p, and those individuals will be slightly mentally retarded, have blue eyes, and very, very light skin. All right. So that's the end of chapter 5. There will also be the last few slides of the full lecture that's posted on Blackboard, has the horse coat color lecture a part of that. So just realize you guys won't be tested on that, but you should, if you're interested in the horse coat color stuff, go ahead and feel free to check